Hello again. So in part one of this, we looked at meiosis I, and we basically said that in meiosis I, what we see is the separation of homologous chromosomes. So in prophase I, these homologous chromosomes pair up, and then they kind of line up in pairs, and they separate out like that. So by the time we're done with meiosis I, we have two haploid cells. Now, each of these haploid cells, though, the chromosomes within them, still have two sister chromatids connecting to each other. So that is where meiosis II comes in. Meiosis II separates those sister chromatids. The process is very similar to mitosis. It's just that we have the names prophase II, metaphase II, anaphase II, and telophase II. So again, it's very similar to mitosis. It's just that we're starting with a haploid cell. And so if you look at this picture, you can see in prophase II, the chromosomes have condensed. Metaphase II, they line up down the center. Anaphase II, those sister chromatids both go in opposite directions. And then in telophase II, they split apart. So I'm not going to go through all those steps in too much detail, but I will kind of show you this main big picture here of how this goes down. So let me go ahead and switch to my other camera. All right, so we left off with meiosis I right here with these two products. So let me just go ahead and redraw those now for prophase II. So prophase II, there's a brief interphase in between these, but other than that, the DNA is unchanged during that. So we have our big, medium, and small for each one, and then I'll go ahead and label those. Little a, big B, little c, big A, little b, big C. And so again, so far our prophase II is going to look just like what we ended with at telophase I. And then again, these are going to line up down the center of the cell. The sister chromatids are going to pull apart, just like in mitosis. And so by the time we're done in telophase II, we have successfully separated the sister chromatids. So this big A chromosome is going to be separated out like that. The big B is going to be separated out like that. And the little c is going to be separated out like that. So by the time we're done, we've got two cells that are the same. And then down here, the same thing. The big A is going to split. The medium B is going to split. And the small c is going to split. And voila. You can kind of see how that works. So by the time we're done with meiosis II, all we've done is we've separated these chromosomes and now we have our haploid cells. So at the end of meiosis II, we have 4 haploid products. All right. So now this would be a great time for you to take a moment to reflect on the differences between mitosis and meiosis. A Venn diagram, a double bubble. How are these different? It really is truly worth taking some time to just kind of sit down and make sense of this. So what cells of the body does each occur in? How many daughter cells are produced? How does the daughter cell compare to the parent? How many divisions occur in each? Where does crossing over occur? There's some great videos linked from the Google Slides, and this one compares mitosis and meiosis, and there's just lots of great stuff out there. So definitely sit down, because again, the similarities are nice, but then it's just similar enough to get confusing. So take some time to look at that, and we'll probably touch base in class about it as well. So now, before we move on, I want to take a moment to just talk about some kind of interesting aspects about meiosis. Because it turns out that it happens differently in males and females. So males, as you know, and you don't need to take notes on this, is just kind of interesting, so I'm sure you're all going to fast forward through it, but I'm going to say it anyway. So males, the process of meiosis happens as part of what is called spermatogenesis, the production of sperm. And it happens in the testes, specifically in what are called the seminiferous tubules within the testes. So the testes have basically like a quarter mile, I think, worth of tubing coiled up within them. And all along the wall of that tubing, meiosis is happening. And while meiosis is happening, that's actually producing sperm at the same time. Now, in males, spermatogenesis happens continuously from puberty until death at the rate of 1,000 per second. Think about that. Like when you're counting like 1001, 1002, 1003, you're not only timing seconds, you're also counting how much sperm the average male is producing. It's incredible. Every time you do meiosis, you produce 4 haploid sperm, as we've discussed, because there's 4 daughter cells that result. And then, oh, there it is. The length of the seminiferous tubules of both testes is about 1/3 of a mile, and then the surface area is about 700 square feet. And so, you know, if you're trying to grasp how could it be 1,000 per second, you have to remember you've got cells in the wall of this thing for about 1/3 of a mile worth of cells doing this process constantly. And so along that huge length, about 1,000 of them every second are just going to happen to be finishing up the process. So lots and lots and lots of sperm are being cranked out by the average male, literally by the second. Unfortunately, though, quality control is poor. Not all the sperm are how they're supposed to be. So this is the typical sperm, but then you got these like deformed, messed up ones: large head, pinhead, 2-head, abnormal tail, tapered head, a bent tail, an abnormal midpiece. So quality control is poor. I guess, you know, in terms of sperm production, just like crank out as much as you can, crank out as much as you can. And, you know, it's kind of okay, because remember, the important thing is the DNA inside of there, not the actual sperm cell itself. And so none of these malformed sperm are going to be effective at swimming, and therefore are not going to reach the egg anyway, so they're kind of out of commission. But that's okay, because it's like you got to keep the assembly line moving, and if you get some bad product, so be it, says the male. The optimal temperature for this process, unfortunately, is about 2 degrees centigrade below the core body temperature. And so that is why the testicles literally hang out in the scrotum outside of the body cavity. And you might think about that and think, well, why on earth would the testicles be there? Why not have them inside the body, like the stomach or the pancreas or the liver or the kidneys? Why does it hang out? And the answer is because sperm production is optimal at a slightly cooler temperature. So basically, that's sort of the trade-off, is we can get a little bit more sperm cranked out by putting them there, so be it. And then, you know, males are equipped with behavioral mechanisms to protect them, because if you ever want to see a male move quickly, just threaten to kind of kick him in that particular location, and you will see a guy very quickly. So, you know, it's a little less protected, but men are pretty on the ball when it comes to protecting that particular part of their bodies. What about females? So in females, egg production is called oogenesis, and it occurs in the ovaries. Men produce 1,000 per second pretty much from puberty until death. In women, it's different. Meiosis occurs discontinuously, which means women produce all the eggs they will ever have, which is 1 to 2 million, before birth. And they're arrested at prophase 1. So have you ever seen those old commercials for Maybelline? Maybe she's born with it. Maybe it's Maybelline. In this case, it's not Maybelline. She's born with it. She's got 1 to 2 million eggs before birth, arrested at prophase 1. And then at puberty, about 300,000 of those remain. The rest die naturally. And 1 egg per month will complete meiosis 1 and be released. And then meiosis 2 occurs after the sperm actually fertilizes the egg. So to contrast this, meiosis occurs from puberty, about ages 10 to 14, until menopause, around age 50, which means if you do the math on that, only about 300 to 400 eggs are ever going to be released. Which means in 1 second, men produce more sperm cells than females will produce eggs in their entire lives. However, eggs are high-quality cells. None of this mass production, you know, garbage junk that the men are doing with the sperm cells. Oh no, this is a high-quality product that is formed. And the reason why this happens is because actually only 1 egg results from the process of meiosis. You might think, well, how can that be? There's like 4 cells produced from meiosis. We just learned that. Well, the way it works is cytokinesis happens unevenly. So in this first cell, when it splits in meiosis I, most of the cytoplasm goes to one product. The other product doesn't really get much cytoplasm, so it doesn't have nutrients, it doesn't have very many organelles, it's just kind of a wasted cell, and we call it a polar body, and it just dies essentially and is gone. And then the second division, the same thing happens. Most of the product goes, or the cytoplasm goes into one of the two products of meiosis, leaving a second polar body. And so basically what this means is at the end of meiosis, even though there's 4 products, 3 of those products are these junky polar body cells, and 1 is this high quality egg cell. So that is going to be really quality. The result of this is that the egg is actually the largest cell in the human body, and sperm is the smallest. And so you can kind of see a little bit of what that might look like. The egg is actually visible to the naked eye. It's 0.2 millimeters in diameter. It looks like a little dot, but you could actually see it on a microscope slide with the naked eye. Sperm is the smallest cell, 300 million in 1 teaspoon, teeny tiny. So the question is why? Why do these dramatic differences exist? And the answer is it's all about reproduction. The egg needs to develop inside the female. It is in the right place at the right time. And so we want a nice high quality egg. The cytoplasm of that egg is going to set the foundation for the entire organism. So we want to make sure it has lots of organelles, lots of nutrients, and all the good stuff that it needs to get a good start. Sperm, on the other hand, have to be made in one organism, leave that organism, enter the other organism, swim through the reproductive tract of that organism, and then reach the egg cell. And so sperm have this incredible journey, and it's quite unlikely that they're going to actually make it. So by producing thousands and thousands and thousands and thousands of sperm, it ensures that at least one sperm cell is going to make it to actually fertilize the egg. And would you like an uplifting story? You are the descendant of that one successful sperm cell. We all are the descendant of that one successful sperm cell that made it and fertilized the egg to become us. So in a way, we're all winners. Isn't that great? I can't think of a better place to end the screencast. So we're going to cut it there, and in the next one, we're going to talk about how this explains the laws we learned about from Mendel.